Good morning. We are Leonardo, Brenes, Dayanna, Vargas, and Fernanda Guerrero. We are students from Universidad, Costa Rica, Vanderbilt University, and Universidad Autónoma de Centroamérica, and we're here to present observation of telomere length changes in the deep space radiation environment. According to the World Health Organization, between 2000 and 2015, life expectancy at birth increased 5.5 years. The healthy life expectancy at birth increased 4.6 years. In other words, people are living more, but not all of those years are healthy years, are quality years of life. A solution to this problem can be found in DNA, specifically telomeres.
What are telomeres? Telomeres are located at the tips of the chromosomes. In humans, the telomere repeat sequence is from 5 to 3 TTAGGG, and this important structure is responsible for protecting the chromosomes during DNA replication by providing additional length during the replication process. Telomerase is an enzyme formed by a ribonucleic acid protein complex, present in the germ cells, differentiated stem cells, and fetal tissues. Telomeres are shortened with each replication, and telomerase restores telomeres, maintaining DNA homeostasis, e.a. the stable and ideal condition of the organism. When telomeres are kept long, the chromosomes are properly protected, ensuring correct replications and reducing the effects of aging such as mutations and excessive apoptosis. Now, some of the telomere shortening related diseases are neurological diseases that are related to pathogenesis of neurological diseases and intellectual disabilities like autism, schizophrenia, Alzheimer, Parkinson, and depression. Another ones are the genetic diseases related to DNA mutation because of the telomeres are too short. Some of them are cancer, diskeratosis congenita, and aplastic anemia. And the other diseases are bone marrow failure, that is hematopoietic dysfunction, and pulmonary fibrosis. This disease is generated to poor telomere segregation. Existing work on telomere lengthening in space is the NASA Twins experiment that was the first project to question the effects of zero gravity and space travel on cellular aging. These results showed that the twin who remained on Earth had no major changes on their telomeres. However, what was impressive was that the telomeres of the twin in space lengthened significantly. This experiment demonstrates that anti-gravity may work as a possible solution to the treatment of diseases caused by short telomeres.
Why is our experiment important? Spirit of Inquiry aims to study telomere biology by observing their behavior in deep space. This basic understanding of the molecular mechanism could lead to a better understanding of the general aging process in humans and the resulting diseases. To our knowledge, there is not enough scientific evidence and knowledge about telomeres being exposed to ionizing radiation in its consequence in the long term. High energy radiation is extremely dangerous since it has sufficient energy to eject electrons for atoms or molecules, especially nucleic acids. The Sustainable Development Goals are health and well-being. Studying how telomeres behave in deep space environment could improve quality of life and understanding the mechanisms that affect aging and diseases. Innovation. This mission will serve as a demonstrator of using proven technologies to quickly develop and run new experiments while accelerating transfer of existing technologies to emerging space organizations. And then reduce inequalities. The knowledge gained can help develop affordable treatments to attenuate the effects of cancer, cellular aging, and other diseases.
Our general objective is to investigate the effects of deep space high-energy radiation combined with microgravity on telomeres using yeast as a model organism. We do this to contribute to future human missions to Mars by deepening the understanding of the actual risks presented by cosmic rays and the high-energy solar radiation present during deep space flight. We also aim to contribute to regenerative medicine by observing telomere length changes in deep space to better understand the aging process, cell rejuvenation, and identify the mechanisms related to cancer growth.
Mission phases are 4. First phase, pre-launch operation. The satellite is delivered to launch provider 6 months in advance of the launch. The second phase, launching causing Spirit of Inquiry will launch as a secondary payload on a vehicle headed to deep space. Some available options could be the SLS, Falcon Heavy, or other vehicles headed to the International Gateway station that will be orbiting close to the moon after 2024. The third phase, experiment operations. Once the CubeSat is released from the dispenser, it will perform a detumbling maneuver using the thrusters to achieve the correct orientation relative to the Sun and Earth. And the fourth phase, mission disposal. When the mission is complete, there could be a possibility of a mission extension that, depending on hardware status and remaining propellant.
The experiment must be conducted away from Earth's magnetosphere, where it can be exposed to a similar environment that astronauts would experience when traveling to Mars. We select a heliocentric orbit because it's a common place for upper stages to be disposed in missions that will travel to the moon, Mars, or beyond. This is especially important because in the coming years, vehicles such as Falcon Heavy, SLS, and vehicles from other countries involved in the Lunar Gateway project will be disposing their upper stages in this type of orbit. We selected a solar orbit instead of a lunar orbit to reduce our propulsion requirements. The sample orbit that we produced had a maximum distance from Earth during the mission of 0.3 AU and a maximum distance from the Sun of 1.04 AU. The overall orbit had a perihelion of 0.99 AU and an aphelion of 1.05 AU.
Now, here are the key performance parameters. The downlink bit rate ranges from 8 bits to 32 kilobits. Telomere observation will be performed once daily over a period of 8 months. The GIST will be supplied with nutrients and radiated after a Spirit of Inquiry exits the Earth magnetosphere. Each satellite will carry 10 microfluidic cartridges. Each cartridge will contain 8 unhealthy short telomere GIST samples and 8 health and normal telomere GIST samples. Temperature within the research payload at 25 centigrades.
The technologies that must be future developed are inserting fluorescent proteins next to telomere sequence and cold gas attitude thrusters. Both of them has a technology readiness level of 9 because they have been tested in successful mission operations.
The mission consists of a 6U CubeSat, weighing approximately 19 kilograms. It will contain all of the systems necessary to run the experiment and maintain the spacecraft in stable conditions. It will be powered using solar panels and use a combination of reactionless and thrusters to maintain attitude control. Communication will be handled by X-band patch antennas, and the telomere experiment will be housed in 4 of the 6 units.
Experiment equipment. For the experiment, GIST will be used to study the behavior of DNA in deep space, especially S-Castelli, because it is known for having regular telomere sequences. This is crucial for simulating the human telomeres. Inserting green fluorescent protein next to these sequences will allow the length of telomeres to be monitored. The protein fluoresces when exposed to the light of a specific wavelength, which is provided by a LED light. To reduce development time and risk, the GIST will be stored and grown with the system developed for NASA's Biosentinel mission. Microfluidic cartridges will contain the GIST, with one cartridge having 16 wells. 8 wells will be filled with unhealthy GIST that have short telomeres, and the other 8 are filled with healthy GIST with normal telomeres.
About the communication equipment, Spirit of Inquiry will use a similar TT&C and communication system used by Prison. Will be equipped with 2 low-gain X-band patch antennas, 2 low-gain X-band patch antennas for downlink and for uplink, and 1 medium-gain X-band patch antenna and 1 maneuverable high-gain X-band antenna.
Our mission will use a combination of sensors to autonomously maintain the correct position and attitude. The onboard computer will receive data from a star tracker, an inertial measurement unit, sun sensor, and with this information, it will send commands to maintain the correct orientation relative to Earth and the Sun. For attitude adjustments, it will use reaction wheels because they provide the precision necessary to point the antennas, and they don't require expending propellant. For power generation and storage, we will generate 73 watts generated from solar panels. The orbit we chose gave us the advantage that there would be no day and night cycle, as it is continuously exposed to the sun. We still have 48.8 watt hours of battery capacity for in case the dedoubling maneuver is not executed correctly, and also as a margin fair in case there's faster than expected solar panel degradation or any other problem with the spacecraft.
The total costs are approximately $192,000 from $1,092,000. This data was approximated by investigating prices in online stores.
To respond to this, our mitigation strategies consisted of maximizing launch opportunities, reducing the likelihood of delays, and reducing the likelihood of component failure, as well as overall increasing the safety margins. The spirit of Inquiry's mission is to expand the existing knowledge about the mechanisms of telomeres by exposing them to high energy radiation in deep space, which is difficult to recreate on Earth for extended periods of time. We'll travel in a heliocentric orbit for 6 months, recording the length of telomeres and radiation exposure. With this data, the risks of interplanetary travel will be better understood, as well as the behavior of telomeres. This could lead to more treatments being developed to treat the effects of aging and improve the quality of life on Earth.